diseases. He is trained in cell culture, stimulation, and analysis of innate and adaptive immune cells using ELISA, multicolor flow cytometry, bone marrow chimeras, protein purification, Western blotting, nucleic acid isolation, recombinant conjugate mapping, linkage analysis, animal handling and breeding, microsatellite genotyping, expert in intravenous, intraperitoneal, and intradermal and aerosol infections in mice. He is also skilled in data mining and big data analysis, including Mouse Phenome Database Ensemble Mouse, Genomes Project, GEO Database, ImGen Database, GWAS Studies, Ingenuity Pathway Analysis. He has also developed machine learning training module to classify positional candidate genes in a large QTL, including optimization of number and size of gene sets, ontology, and mouse phenotype terms in GEO expression datasets. He has published many research papers in the world's renowned journals like World Journal of Hepatology, Communications Biology, FESEP, Genes, Genomes, Genetics, etc. So ladies and gentlemen, I am inviting our excellent speaker, Dr. Abbas Raza. Over to you, sir. Sir, your mic is muted.
Let me open it. Sorry about that. So just a second. Can you all see the slides?
Yes, sir.
Okay. All right. For some reason, my screen is just going to the next slide. Yes. Okay. Okay. Thank you, everyone, for the kind introduction and the opportunity to present my work here in this forum. So I'm going to give you a brief introduction on an autoimmune disease today that has a strong genetic component, and it's the disease that I studied for almost like 6 years when I was a PhD student at University of Vermont. And I try to give you like a snapshot of the place where I worked, and it's a very nice university with lots of like good immunology and genetics lab. So if anyone is interested, I'll be happy to provide them like additional details. So this disease that I'm going to be talking to you, it's the #1 leading cause of neuronal disability in young adults. And if you haven't guessed this disease, let me just give you a brief overview. So this disease is called multiple sclerosis, or MS, and this targets the CNS, which has two components, the brain and the spinal cord. And if you look in the CNS, there are like a billion neurons, each having a protective coating of myelin sheath. And these neurons allow us to, do all the physiological functions that we do: walking, talking, speech, cognitive functions. And in an MS patient, if you look at these same neurons, this myelin sheath, which is made up of lipids and proteins, this is actually damaged. And this damage is also apparent when we look at the MRI scan of these patients. So this is a MRI for a MS patient, and you can compare it with a healthy individual. These regions which have these plaques or multiple sclerosis plaques, these light up in the MRI. And hence the name of this disease, that you see these areas of demyelination in multiple areas of the brain. And because of this damage, the neuron is unable to do its like proper physiological function. And you can see here that the impulse firing is actually dysregulated. And since this can happen throughout the CNS, and since each of the parts has a different function, the symptoms are also very variable. So you can have hearing loss, double vision, slurred speech, cognitive dysfunctions. And in 50% of the cases, like once the disease is diagnosed, like within like 10 to 15 years, they are like on the wheelchair. So it's a really bad disease to have. And since it targets the young adults between the ages of 18 to 30, you can imagine like living with this disease for the rest of the life. There is still no cure for MS. And so most of the therapies that have been developed, they are trying to reduce the symptoms of MS. Like there are a couple of these which I've mentioned, and I'll just provide more information. If you look at the clinical forms of MS, There are four clinical forms. So for example, I'll just give you a brief introduction on this form, which is the most common form of MS we see. Like more than 80% of the individuals, they have the relapsing remitting MS. So this is a score of disability of the individual with respect to time. So you can see in this case of MS, there are periods of symptoms like where you see symptoms show up for MS and then they disappear. And then again, there's a period of relapse. And gradually, the disability also goes up. So these individuals, when they're undergoing these symptoms, you can imagine they could be on wheelchair or they can't walk or speak, but then they recover and then the cycle repeats. The age that these different forms target is also different. So for example, relapsing remitting is like in 20 to 30 years. If you look at primary progressive, where you see symptoms show up gradually and they don't disappear, this is mostly targeting old people. And also the therapies that have been developed to like improve these symptoms are also different for each. So you can imagine it's a complex disorder with complex therapies to treat. Looking at the epidemiology of MS, so where do we find these cases of MS? When I was in Pakistan, I had not heard about this disease because it is not so common. And part of the reason is also like there's little diagnosis of MS as well. But like recently, I've seen papers where people have looked at the MS cases and the incidence has been rising even in like the Indo-Pak subcontinent. But overall, if you just look at this graph, you can see like most of the cases like are found either like countries like the US or Canada or the European countries, or if you go further away from the equator, not just the north side, if you go in the southern hemisphere, even Australia and New Zealand, they have like a high incidence of MS. And some of the countries, they don't have data available, so we don't know. This disease is slowly climbing up. Like now we know that there are 2.5 million people affected. In US alone, there's a million people. And this is the incidence of MS in like other countries per 100,000. So India, Pakistan lies somewhere here. And I'll talk to you about like the risk factors, what makes individuals here less susceptible to versus say in England where there's a lot of incidence. Okay, in terms of the pathogenesis, so how does it happen in terms of the immunology of MS? So if you look at these lesions which light up in the MRI, there's a lot happening. And so this is the blood-brain barrier that separates the periphery from the CNS. It has been known for a long time that there has been activation of immune cells, specifically targeting the CNS antigen myelin. So one of the cell types important is CD4 Th1, and so these are pro-inflammatory T cells. So once they gain access, we don't know how, but once they gain access through the blood-brain barrier, they recognize this self-antigen and start secreting a bunch of cytokines like interferon gamma, IL-17, which recruit additional immune cells like macrophages or B cells. They further go in, start killing the myelin sheath, and this vicious cycle keeps on repeating itself. So more immune cells come in, they degrade the cells in the brain oligodendrocyte. They try to relieve the symptoms by making more myelin, but after a while, the symptoms become so severe that ultimately you have this paralysis. Now, moving on to the risk factors for this disease. And we know that we can characterize them into three categories, like sex of the individual, the environment where the individual resides, what he eats, what he does, his physical activities, and also the genetics. And most of my talk will be on the genetics, but I wanted to give you a snapshot of all these risk factors. And all these interact as well. So for example, if you look at a particular ethnic group that has a much higher incidence of MS, like say people from the Persian ancestry, depending on in which environment they are, their risk could be different. So if they are in a susceptible environment, which I'll talk about, they get MS, but if they are in a resistant environment, they don't. And also MS is much more common in women. So let's first start with the risk factor, which is the sex of the individual. So historically, it has been known even when MS was first described back in 1800s, that there were much more women affected by MS than men. If you look at the white Caucasian population, the ratio is 3 to 1. So there are 3 times more women than men that get diagnosed with MS. If you look at African Americans, which is also a big chunk of the population here, the ratio is much more skewed, even 6 women to 1 man. So clearly there is a sex component. And not just the sex of the individual, just having like differences in the hormone, especially when it happens during pregnancy. So this has been seen that if this is the relapse rate for MS, where the symptoms show up, the symptoms drop during the third trimester of pregnancy, which suggests there is an important contribution of hormones. And then they return back after delivery of the baby. Conversely, even though men don't get MS quite a lot versus the women, the ones that do get MS, their symptoms uh, it's much more rapid. So you can imagine like men getting like on the wheelchair much earlier than women. So there's a faster disability accumulation, which is also like interesting. Like there are differences in the disease outcome between men and women, and also in incidence. And we also know from studies, like I mentioned a couple of them, like testosterone is also linked to like the levels of this hormone is linked to the disability. And even if we just exclude the role of hormones, just having like the X and Y chromosome can have an effect. So our lab has actually published on this, that depending on the Y chromosome, in the animal model we have, depending on the Y chromosome they inherit, the outcome of disease could be very different. So it's a very interesting like area, and I work quite extensively on this area as well. So moving on to like what else? So here I listed like a couple of more things that could affect like how much exposure you get early on in the life that can affect the outcome, but we're not going to talk about this. So moving on to the environmental risk factors. So we know like if we plot like the incidence of MS or the prevalence with respect to the latitude, so how far you are from the equator, the risk goes up substantially. So this is towards the southern hemisphere and this is towards the northern hemisphere. And you can see like countries like at the far end have much higher MS cases. So this clearly speaks for like the environmental component of MS. One of them, which has been shown quite a lot, like there have been a lot of studies, is vitamin D levels. So we know that the vitamin D level fluctuates depending on where you are on this latitude like line. There's also an important contribution of diet. So we know vegetarian diet reduces the risk for MS versus the American diet, high fat, sugar, salt diet increases the risk. We also know that exposure to pathogens can also affect the outcome. Smoking is also a risk factor. And also recently, microbiome was also shown to be important in mediating the risk for MS patients. But I'm mostly interested in the genetic component, and this talk will focusing on that. So let's just talk about the genetics of MS. So when MS was first diagnosed, it was known that it runs in families. So if you look at the familial risk for MS, like if you have one individual in the family, what is the risk if you're in the family for like another individual that you will find for MS? So the risk is quite high versus the general risk is like 0.4% of the population gets MS. And not only that, if you look at particular racial and ethnic groups, so MS is much more common in like people from Scandinavian countries, British Isles, North America, Persian ancestry, and African Americans. But MS is very rare in like other communities, especially like Indian Pakistanis that I mentioned. So that speaks for a genetic component. The gold standard for these genetic studies was done looking at the monozygotic twins. So looking at just identical twins and comparing if one of them has MS, what is the percentage like the other individual also gets MS in their lifetime. So we find like these studies, they predict like 24 to 30% risk. So clearly the genetic component is like 30%. However, if you look at dizygotic twins, so they don't share like 100% of the genome, the risk goes down. And if you go further down and down, so say me versus another individual, the risk is very low. This speaks for a polygenic mode of inheritance. So it's not just one gene contributing to MS risk, but there are several genes. And if you add in the risk for across all these genes, that could explain the 24 to 30% genetic component. So initially, when people looked at these monozygotic twin studies, they were like, we want to map the genetic component. So they went ahead and did linkage analysis. So taking a family, trying to look at polymorphisms that segregate the susceptible from resistant individuals. They found like back in like 1990s and 2000 that the highest like the gene that showed up the most was the HLA class 2 or the MHC class 2 that we know. And it was giving like a strong penetrance. This molecule, we know, like if you look at the immunology, it's important in antigen presentation. So it makes sense. A T cell that can recognize this polymorphic MHC molecule much more strongly should be able to respond more effectively and cause disease. So that was like in agreement with what we had, the idea that these immune cells are important in MS. However, later studies that were done looking at the same like bigger and bigger families, they did not find or uncover any additional genetic variants. So that was something like people wanted to pursue, but they could not find. So around that time, the human genome was also sequenced, and we also had data from the HapMap project looking at genetic variants in the human population. So then the thinking changed. Maybe MS is not like cystic fibrosis, where you have uh, genetic variants that are very rare in the population, but they have much higher penetrance. But in fact, MS could be caused by common variants with very low effect sizes. So you can see like 1 or 1.5. And they are much more common in the population, and each of them is contributing to like some part of the risk. So in order to answer this, we wanted to do like much larger studies. And this is essentially what researchers did. They went ahead and did genome-wide association studies, or GWAS. So what these studies do is they take in almost like 10,000 to 20,000 cases of MS and sequence the SNPs. So trying to distinguish the SNPs that can segregate maybe MS cases from, say, healthy individuals or controls. And because these studies are done with several thousand individuals in each category, it has much more power to uncover these variants. So for example, one of the study here, which I've highlighted, they look at all the chromosomes in the human and then trying to map which variants show up as much more significant in the MS individual. So you can see these are the peaks that come up. And because of these scans that were done in the human population, we are able to identify almost like 230 SNPs or genes that are associated with susceptibility. These are some of the studies that have been done, and because these are so expensive, like one of the studies takes like $1 million. It is very important like we understand fully like what that data means. Most of these predicted variants in MS, they have immune functions, so that also supports the idea that MS is an autoimmune disease. And they're also shared across other autoimmune diseases like lupus, and we have several other examples. That speaks for the shared like pathway of these diseases with other autoimmune disease. However, even though we have like done all these GWAS studies, we know about these genetic variants, still we are limited by a couple of like issues here. And I'm going to highlight them. And this is the basis of my thesis. Like I try to validate these GWAS studies. So for example, if I add in all these genes that have been predicted by these 10, 12 GWAS studies, like all these 233 variants, they explain only 20% of the genetic component. The rest of the genetic component is unknown. So that is strange. If all these studies have uncovered is just 20% after like 10 years of GWAS, then why are we missing this 80% of the genetic component? Also, these GWAS studies, the SNPs that have been identified, most of them are like in the non-coding region. So like introns, regulatory elements, or pseudogenes in the human genome. We don't know which gene it targets. So we are missing a lot of the information there. And even if we just look at linkage disequilibrium blocks in the human genome, so if this is the human chromosome, you can see like it is segregated into these individual early blocks where you have a SNP that was genotyped in the GWAS chip, and this is another SNP that is very close to the GWAS chip, but actually is the true causal SNP. When you are looking at segregation of these two SNPs, they will segregate together. So you may miss this SNP, and in fact, you will assume like maybe this is the causal SNP. So we need to take into account that these LD blocks and the causal SNPs. And because the LD structure varies across different populations like Caucasians, African Americans, a finding from a GWAS chip may not apply to across all these ethnic groups. So we need to take that into account. Also, these GWAS studies, they're excluding rare variants. That was the whole idea, like just look at common variants. So we are missing a lot of the heritability from that part as well. In fact, a recent GWAS study that was done exclusively using these rare variants, they uncovered almost like 5% of the genetic component. So now we know 20% plus 5%, like 25% of the genes contributing to MS. And then there are, as I mentioned, there was several different forms of MS. So each of these are different, like relapsing remitting MS is different from primary progressive MS. Since these GWAS studies, they just look at broadly, irrespective of the clinical form, what are the genetic variants. We don't know whether a particular variant is important in, say, one clinical form over the other. And lastly, because these GWAS studies, they heavily involve typing variants on the autosome, so not the X and Y chromosome, we don't know a lot of the information from there. So we are lacking those variants that are found, especially on the Y chromosome. And part of the reason is we don't have good polymorphic markers for this. So that is the limitation. And because has a very strong sex bias, we may be missing a lot of the information from these GWAS studies if we exclude X and Y chromosome. So having said that, if you just look at the GWAS study, it's an association study. It says genome-wide association. So it's not a causal study. So we don't know exactly, does it really cause the disease or not? So the biological function of most of these loci that have been predicted for MS, we don't know. And it is very important that we perform mechanistic studies. The problem with human subjects is you can't just take a person's brain and chop it off and look at the individual cells. So it has to be done in, say, animal models, which are suitable for MS. And even after like we are able to find, say, postmortem like brains to study these genes, the contribution of each GWAS hit is so small. So I told you like the penetrance was like very low. Studying the role of each of these loci in pathogenesis of MS and also like segregating the sex and the non-sex specific mechanisms, it's very hard to do this. So this is the reason like we have pursued like validating the GWAS hits using a mouse model. And this is the mouse model that has been used in MS for quite some time and is the principal animal model, which we call experimental autoimmune encephalomyelitis or EAE model. This model works by immunizing a set of susceptible inbred strains of mouse with a myelin antigen. So this is the CNS antigen where the immune response is generated. We inject it twice at day 0 and day 7. And then we follow the disease in these mouse over the period of like the next 20, 30 days. So in this model, we are trying to gauge these clinical features, which are scored here in this 0 to 5 scale. So if the mice remains healthy, which has no clinical signs, we give it a score of 0. If the mice develops a floppy tail, you give it a score of 1. If they subsequently develop paralysis and like further extensive paralysis, you can see the score goes up. So this is one way to actually look at the clinical form of like the clinical like severity of disease using this animal model. And there are a couple of things we can look at. We can look at the disease early on, what's happening. We can look at the peak of the disease, which is when the immune cells enter the CNS. And we can also look at the recovery period when the symptoms are, disappearing a little bit. So after this 30-day period, what we essentially see is a plot of like the clinical score with respect to time. So for example, if these are two different strains, B6 mouse, which is the common strain that we use in the lab, and this is say another strain. Just looking at these two graphs, you can see like the B6 mouse is much more susceptible. So it reaches a score of 2, and the C1 mouse is probably resistant. So it never reaches a score of 1. Only some of the animals do. And since we are averaging from several mouse strains, it remains below 1. And you can also see like the disease starts around like day 14 for some of them. The peak is reached around day 22, and then this is the recovery period. So this way you can actually assess different inbred strains. Now coming to, since we are trying to model this disease in the mouse, there has to be like some homology between the disease and in the human and also the disease in the mouse model. So here I've just listed a couple of things just to give you an idea, like EAE is no more different from MS. First, EAE is also has a genetic component. So there are some strains of mouse which are susceptible and some strains which are resistant. Just like in the human population, there are some ethnic populations which exhibit a lot of susceptibility. And also EAE has a lot of genes contributing to its susceptibility. So it's a polygenic disease. We also know there are sex specific. So the disease that we see in the female mouse is much milder than the disease that we see in the male mouse, just like what we see in human population. And each of these genes or the loci in EAE also has a modest effect. So that makes sense. Also, in terms of the pathology, EAE is similar to MS. So there are CD4 T cells important, and then there are other immune cells. And most of these drugs that have been developed, or the therapies that I mentioned for MS, they were validated first in the EAE model. So you can imagine like it's a very good model to study MS and develop therapies. So now coming to the focus of my research, we wanted to test a bunch of these genes called the SLAMs, which is short for signaling lymphocytic activation molecule family, or SLAMF. If you look in the location of these genes, these are a family of 9 genes, which are shown here from 1 to 9. And they're highly conserved between human and mouse. So this is chromosome 1, and they're both found on chromosome 1 in these two like genomes. It's highly conserved, which suggests it has an important contribution, and that's why it has been kept intact in these two genomes. It is expressed on all hematopoietic cells. So B cells, T cells, macrophages, dendritic cells, they all express these SLAMs. And these are the different SLAMs that are expressed. They have very important immune functions. So these are important for co-stimulation of these immune cells. So CD4 T cells stimulate differentiation, proliferation, activation, natural killer cell functions. And importantly, these SLAM family members, they came up as possible GWAS candidates in MS studies. So SLAMF 1, 2, and 7 was predicted in 3 GWAS studies that I know so far. And not only that, it is also implicated in other autoimmune diseases like lupus, and I just listed a couple of them. So we thought this is an interesting family of genes that we need to verify. Are these really the causal loci in MS or not? So we looked around to see, is there a way to model this in the mouse? And in fact, back in like 2003, this group at UT Southwestern from Wakefield, they actually had sequenced the SLAM congenic interval across 34 inbred strains of mouse. And they had found that this locus, even though it's polymorphic, it segregates into broadly 2 haplotypes. So we have a bunch of these strains that have a different makeup of the SLAM genes. So B6 is very different from this haplotype group, which has 129 strain. So we thought this is a natural way to test the effect of this polymorphism, just like we do GWAS studies by taking cases, looking at their SNPs, comparing it with the control. if we can take a B6 mouse, compare it with 129, we have a way to test this. And our job was made easier because there was a congenic mouse available that had captured the locus of SLAMs from 129 on a B6 background. So this is the mouse that was developed. So it had a different polymorphism, like the 129 integressed on a B6 background. So when we use this, so this is how it looks like. So we have a C1 congenic mouse. The background is B6, and only the SLAM locus is different. So C1 has a 129 interval. So all the other genome is B6 except for the SLAM locus. And we wanted to compare this with B6. And the hypothesis was, if this is different from B6, it means the SLAM locus is important. And in addition to that, we also made use of another congenic, which has a congenic mouse, which has a smaller interval of 129. Importantly, they both have the GWAS candidate that I talked about. So SLAMF 1, 2, and 7, which were predicted to be important in MS in these GWAS studies. And this was developed by like a lab next door to ours at UVM. So we made use of that as well. And we compared it with the B6. So now I'm going to show you like how much these two strains differ, like the 129 and B6. So if we look at the SLAM locus across these, the two strains that I'm using, B6 and 129, all the SLAM members are different. So these are all the SNPs that distinguishes the B6 and the 129 strain. And the C1, which is the larger congenic, has all 9 members, and C2 has like members including the F1, F2, and 7, which are the GWAS candidate. So now I'm going to show you the results that we saw. So this is the EAE graph that I showed you earlier. So mean clinical score is on the y-axis, and the x-axis is the time post-immunization. So just looking at these black like circles, or the fill circles, these are the B6 mouse. So they are susceptible, as you can see. If you compare C1 and C2 congenic, they're also susceptible. And we don't see any difference. And this is true for females. So we thought maybe the SLAM locus is not important in the females. However, if we look at the male mouse, we found this interesting phenotype that the C1 congenic, which has the larger SLAM locus, they were resistant to EAE disease. And so they reached somewhere like around 1 score versus B6, which were susceptible. Moreover, the C2, which was our positive control, because this has the shorter interval and also has the GWAS candidates, this was susceptible. So the phenotype was in this locus, but not in this locus. And there are a couple of like the stats that we have done, but I don't have time to go over these. But we found like the C1 was clearly like resistant across different parameters, like incidence of the disease, peak score, day at onset, and severity index, but I don't have time to go over those. So clearly the causal gene, or the loci, or a group of loci that we believe lie somewhere in this distal locus. We also know it's sex specific because we see this phenomena or phenotype in the male mouse. And we also know from these, comparing these two congenics, that it's not the GWAS candidate, the F1, F2, and F7. So what else do we know about the C1 congenic? So we did immune profiling, which is essentially taking the immune cells at day 10 or day 22, running them on flow cytometer. So looking at different immune cell population important in EAE disease, it's like CD4 positive T cells, CD8 positive T cells, CD11B, and CD19 are the B cells. We also did immune profiling. In addition, we also looked at the ELISA from these lymph node and spleen that we took out from the mouse. And we looked at different cytokines important in EA disease, like GM-CSF, interferon gamma, IL-17, and IL-10. And we did the same both in initial on in the disease and also later on, like in the CNS, which is the peak of the disease. Just walking you through some of the results. So this is the draining lymph node at day 10. And I just want you to focus on only these three charts. So the immune cells that were much higher exclusively in the male population, this was CD11B positive, FOXP3 positive or CD4 positive, FOXP3 positive, which is also the regulatory T cells. And we also found CD8 positive, IL-17 positive, much more in the male. And only these populations showed up, whereas the others were not significant, or they did not segregate with the male phenotype. So we also confirmed this in the CNS as well. So just focusing on these two populations. In the CNS, also the Treg population was much higher in the male versus in the female, it wasn't significant. And also if you look at this population, the CD8, it was much more significant in the females. So clearly the only population that shows up segregating with the male phenotype is the Tregs. That could explain that why we were seeing much milder form of disease in the male mouse, that maybe these Treg populations are somehow regulating the immune response. We further validated this by using the supernatant and running ELISAs across these different cytokines, which are shown here. And just focusing here, the only cytokine that was significant was the IL-10, and it was exclusive in the lymph node. So the males were showing much higher, they were secreting much more IL-10 when we did this assay. Okay, now that we have confirmed some of the mechanisms, uncover some of the mechanisms, how these mice are resistant, I wanted to validate the male specific phenotype. So I did orchiectomy experiments, which is essentially you take the congenic mouse, perform orchiectomy. So you remove the source of the hormone and also the organ, like the testes, and then you do the reverse. So you supplement them with either empty pellets or control pellets or with hormones. So these are the two male hormones that we have used, testosterone or dihydrotestosterone. The difference between these two is DHT is non-hydrolyzable, whereas testosterone we know can be converted into estrogen. So we wanted to segregate the phenotype over there. And then looking at what happens to the EAE disease. And as a control for all this experiment, we also did sham surgeries, which is essentially you do the surgery, but you don't cut off the organ and then just suture the animal back and then do these experiments. So let me just walk you through what we saw. So this is the clinical score as before. This is the day post immunization. C1 is the original congenic mouse resistant, as I showed you earlier. The sham surgery did not do anything. They also remain resistant. Looking at the orchiectomy, C1 orchiectomy with the control pallet, so this one, the CNT1, you can see the phenotype was lost when we perform orchiectomy. This confirmed that this was a male-specific phenotype, and by removing the testes, the phenotype became as susceptible. So it's the same as the B6 I showed you earlier. Interestingly, what we were not expecting was when we supplemented with DHT or testosterone, which we were hoping would return back to the same C1 level, we found it did not recover the phenotype or restore the phenotype. These animals were as susceptible as if they had the orchiectomy done with control pellets. And this suggests maybe this phenotype that we are seeing in the C1 males, it is dependent on the testes, clearly, because we lost the phenotype, but it is not dependent on testosterone or DHT. And as a confirmation, we also have ELISAs done to looking at the testosterone levels and DHT levels in these supplemented strains. You can see the supplementation was fine, but we did not recover the phenotype. Interesting. And we believe that because testes are making so many secretions, there could be other ways that this phenotype could be regulated. And there are endless secretions that we can perform these supplementations, but we did not go into that route because we don't know exactly which secretion to use. Okay, now that we have established that the congenic C1 has the phenotype and C2 don't have the phenotype, which is shown here, Next, what I did was I made another conjenic line, which is essentially spending hours and hours in the lab and crossing these mice and hoping I can get a mouse strain which captures 129 region distal to the SLAM, like the SLAM F1 to 7. So we made this C7 congenic, and we found this was susceptible. So it is capturing, you know what? I think I wrote this down. This was resistant. So C7, which is the region distal, it was able to capture the same C1 phenotype, and I apologize, this is resistant. Then I also made additional congenic lines, furthering, like narrowing down this locus more and more. So these are the different regions that were captured from 129 on the B6 background. And just to orient everyone, this is the SLAM GWAS candidates, F1 to F7, and these are the other SLAM members. So they're found in the C7, but not in the C8 and others. So when this is the result that I mentioned. So if you look at the EAE disease with respect to time, C1 and C7 are as resistant, suggesting like the loci lies somewhere here in the C7. But C8, you lose the phenotype, and also in C10, C13, and C11. So going back again, if you lose the phenotype in C8, 10, 11, 13, this suggests the region that has the gene for this phenotype lies somewhere here. So this is also highlighted here. So this is our region which has the candidate. And in fact, it has the true causal GWAS variant. And it's a much smaller interval compared to the 5 megabase pair that I initially started with. And we also know it's a recessive trait, as shown here. So then I asked, is there any way I can narrow down this region? So this is the region that we are interested in, which is in C8, but not in C7. So there are several genes. So we asked, are any of these genes dependent on the testes? So we looked at gene expression data sets. One example of this you can find in the GEO database. And the cell type that I looked at was a CD4 T cell and granulocytes. And I asked, is there any gene that is affected following orchiectomy? Because this could be the gene that I'm interested in. Excuse me. So we looked at the genes, and only 6 of them came up as different following orchiectomy. So ATP1A2, KATN3, and then there were like 2 other SLAM members as well, F8 and F9. The arrows show either they go up or down, and these are the SNPs that can distinguish 129 and B6. And because these genes have important immune functions, these are our list of candidates that are important in our EA model and could be the true causal GWAS variants. Overall, what I have shown you in this study is how important it is, like these GWAS findings that are published, they are taken with a grain of salt. They need to be validated. And when we validate these GWAS studies, we find that in fact, it wasn't the GWAS candidate, but a gene and linkage to the GWAS candidate that seems to be important. And I'm not saying it's SLAMF8 and 9, I'm just saying all these 6 genes could be possible candidates. And because our study focused exclusively on the male phenotype, we believe like once we narrow down the candidate, we may be able to target MS exclusively in the men, which we know have a different phenotype or clinical form than women. So if I summarize the finding, what we have found is through congenic mapping, the loci that contributes to the CNS autoimmune disease, it lies distal to the SLAM locus, like the SLAM GWAS candidate, the F1, F2, and F7, and it's male specific. I've shown you the immune population that is important for this phenotype in the male mouse. I also showed you through orchiectomy that this was a male specific phenotype. And when I did orchiectomy, the phenotype was lost. And when I did hormone replacement, the phenotype was not restored, suggesting it's testis dependent but not hormone dependent. And through gene expression datasets, I am able to narrow it down to 6 candidates that are differentially expressed, and their expression is testis dependent. And we will be able to use these candidates once we validate them to develop therapies for MS in men. So in future experiments, since when I left the lab and I started my postdoc, the lab has been focused on doing like follow-up experiments in these mouse congenic strains that we have developed. So we are doing immune profiling or characteramine hormone replacement in the smaller congenic lines like the C8 and C7. We're also interested in looking at the biological interactions with the testicular secretions and the genes that we narrowed it down. So we know like the genes that are expressed in the testes. So just looking at what are the possible interactions. We're also validating these 6 candidates using the RT-PCR or expression of these genes. However, one of the caveat is we don't know the cell type that is important. So we're looking at a bunch of these cell types, especially the Treg population that I mentioned. And also validating with the CRISPR-Cas9 will be the ultimate goal. And that will be a confirmation that this gene is really the causal variant important in this disease. So with that, I would like to take a moment to thank people in my lab. So this is the group where I work with whom I work with at UVM, my advisor, Corey Tusher, and all the members of the Immunobiology department. I also want to thank the current lab where I'm working at, UMass Medical School, where I'm looking at like another disease like tuberculosis. And if you have time, like later on, I will be happy to talk about that. But with that, I'll be happy to take your questions. And if you have any comments, please feel free to either put it in the chat box and I will be happy to answer them. Thank you.
Thank you so much for this enlightening and wonderfully informatory webinar. Proceeding to the question answer session, I have this question that how gut bacteria microbiome is involved.
Yeah, so that's a very interesting question. And we know like several studies have been done using the gut microbiome, say from a mouse strain that was susceptible, taking out the gut microbiome and transplanting it into a resistant strain. And we have seen that this can impart susceptibility to the resistant strain. And the same is true in the human population. We know like there are a subset of gut microbiome which are different between MS patients and healthy individuals. And so how it does that, the question is how it is done. So these gut microbes, they make several metabolites like within the gut, and several of these are important immune modulators of these immune cells like CD4 T cells that I mentioned. And I remember like one of the studies, they looked at Prevotella, a strain of Prevotella microbes, like the genus, and they found it was protective. So this is how they modulate the disease, by providing these metabolites that are immunomodulatory. I hope that answered the question.
Thank you so much. We have another question that why are most autoimmune diseases more common in women?
This is my favorite part of the talk, like why are there sex differences and why we see it much more common in women. So I won't say like all autoimmune diseases are biased towards women. There are some diseases which are much more common in men. And so one of the reasons is like the hormone influence. So testosterone and estrogen. So testosterone, the male hormone, it's immunosuppressive. So if you look at individuals that have much higher levels of testosterone, their MS symptoms are much milder versus if you look at individuals. So let's say older individuals have much more severe disease because the testosterone level drops. In the case of estrogen, the immunosuppressive properties are much milder. So that's why you see the disease is much more common in women. And also, not only just the hormone, I told you like the X and Y chromosome itself has a role to play. So the genes that are expressed, we know in the animal model, by having different polymorphic regions in the X chromosome, it can have a different outcome than the Y chromosome. But since we haven't done a lot of these GWAS studies, so we don't know exactly what happens in the human population.
Okay, thank you so much, sir. We have another question from Manahil Beg. Is there any specific reason that MS is more common in certain ethnic groups like African American population?
Yeah. Yeah. So this is the whole idea about the genetic component, that we see like these different ethnic groups have different MHC haplotypes. So one of the genes that I mentioned was the strongest signal for GWAS studies was which haplotype of MHC class 2 one has. So in some ethnic populations, there are polymorphisms that are much more prevalent. So you can imagine their immune cells are much more hyperactive, so they respond much stronger to the myelin antigen or the self antigen. And so we see like in the African Americans, the particular variant of MHC is much higher, which is associated with susceptibility, and vice versa, say for a resistant population like Indian and Pakistanis.
Okay, we have this last question that in your study you had different strains. One was C112 and the other was C1 sham, which had undergone this mock surgery. So there was a slight difference in the results among C112 and C1 sham. So can you explain the reason for that?
Yeah, so one of the things that we look at, like when we're doing these experiments, is how different they are statistically. Like, is there any difference when you compare these two? Of course, when you're doing sham surgeries, it won't be as identical as like the C1, which you haven't, done any surgery on. There was very minute difference. So you can imagine the clinical score was zero point 0.8 for the sham surgery or 1 for the like no surgery control. So they are no more different. Like when you're looking at compared to say a B6 mouse, which had a score of like 3. So even if you see those milder differences, they were not statistically significant. So we exclude them. The only difference that I could see is their incidence. But since we are looking at different parameters like incidence, the cumulative disease score, if you add them up, they are no significant. I hope that answered the question.
Yes, sir. Thank you so much. I guess these were the questions. And thank you so much for joining, everyone. And especially special thanks to you, Sir Dr. Abbas, for enlightening us with such good information.
Thank you for inviting me. Yeah. If you have any more questions or anyone wants to, reach out to me, I'm happy to answer more questions. And I think I'll be happy to share my email. It should be on the flyer, but you can shoot me an email. I should be available. Thank you.
Sir, we have another question.
Mhm.
Uh, why the testes are involved and the HTD and testosterone?
Yeah. So I mentioned like we normally we believe like testes, most of their physiological functions that we see is due to these male hormones like DHT and testosterone. But testes are making several secretions, and I did not mention them, but there are inhibins, activins, and they're like 200, more than 200 secretions that testes are making, and they have important biological functions. Since we are not looking at them, like we did not test them, we don't know exactly which of them contributed, but the ones that we tested was the DHT and testosterone, and they did not. And I remember like when I was doing this experiment, looking at like, is there any other secretion important from the testes? So we do see like interaction which are mapped in like bioinformatic like databases from like inhibins and activins with the SLAM interval. So these could be important. And one thing that I wanted to mention since this question was brought up, there was a study done back in like 2018 looking at testicular environment and what it does to the immune response. So historically, it has been known that testes are like they're immune privileged organs. So if someone inject antigen into testes, like a foreign antigen, there's very little immune response generated in the mouse model because it's so immune privileged. There's a lot, not many immune cells going into the testes. But in that paper, that showed that in fact within the testes there are macrophages, like a specific kind of immune cell, which are immunosuppressive. So rather than being like immunoinflammatory, they were immunosuppressive. And just having the environment itself can affect the outcome or the phenotype of these immune cells. So that is also another hypothesis that by irrespective of the secretions these testes are making, just excluding the testes could affect the phenotype because it's so immunosuppressive.
Okay, thank you so much, sir. I'd like to invite Dr. Muhammad Ali to say a few words.
I think I don't see Ali in the Zoom.
Assalamu alaikum. Thank you so much, Dr. Saab. Thank you
Assalamu alaikum.
for such an excellent talk and for the time, especially at your 6 p.m. And Dr. Saab, we have faculty like Dr. Mureed Hussain and Dr. Asma Irshad. They are very good human geneticists, and we look forward in collaborating with you, like in this project and specifically in the sample collection and in this type of thing. So we look forward to you if we have any, we can have any some meeting regarding that. So
Thank
we
you,
will look
Ali.
happy
Thank
for
you for
that.
the
Yeah.
invitation. Yeah, I really enjoyed like interacting with everyone, and yeah, I look forward to interacting further with the faculty. Thank you.
Thank you so much. Thank you.
Yeah, hope everyone have a good rest of the weekend. Thanks. Bye bye.
Bye bye. Allah Hafiz.